So protein is a biomolecule that have different function. On if we try to classify the protein, there is a two type of a protein. Very roughly we can classify or differentiate. One is the structural proteins, or those have the, and another one is the functional proteins. Structural proteins, what they used to do, they generally make the or keep the structure or the framework of the cell or different type of a organelle, and they have a this type of a function. They actually participate for the structure, and the function vastly belongs to the enzymes. Now how protein form? So for protein formation we need amino acid, and to understand the structure of a proteins we have to understand from the basic. So proteins structure, it is a kind of a hierarchy. So it can be divided into three categories, like primary structure, secondary structure, tertiary structure, that is the major one, and another one that we call them as a quaternary structure. So primary structure, from the primary structure we just able to understand the sequence or arrangement of a amino acid. It is a long chain of a amino acid. So both two amino acid is connected by the peptide bonds. So if for example any proteins, they have a 300 amino acids, so that 300 amino acid came together and stay together with the help of a peptide bond. So here the primary structure completely depending on the peptide bond. The other bonds when participate, then they make the secondary structure. Secondary structure two types. One is the alpha helix and another one is the beta sheet. Now alpha helix, to for formation of the alpha helix there is a specific group of amino acid if present in a primary structure, so they are have a tendency to make a spiral structure, so we call them as a alpha helix. Definitely alpha helix have different characteristic. One of them is the like DNA helix, so it's also a right-handed helix. And the beta sheet means two amino acid chain, they will come together side by side to make a beta sheet. Now beta sheet also two type, one is the parallel, another one is the anti-parallel. Parallel in the sense both of these amino acid will face the same. Like if we consider the C terminal and the N terminal, C terminal in the sense carboxy terminal and the N terminal in the sense amino group terminals, right? Like we know the amino acids, the structure in that amino acid structure, there is a one aromatic carbon. So in the, on that carbon, one group attached with the hydrogen, another covalent bond attached with the amino group, another covalent bond is attached with the COOH group, and the last one is the R group, like this. So that COOH group is the carboxyl group or the C terminal end, and the amino group is the N terminal ends. And we used to count from the N terminal amino acids. Okay, like for example, if it is started with the methionine, so it will be the first one and it should be the N terminal side, and the last one if it is lysine, so there will be the carboxyl group will be the free, they will not engage with any other amino acid, so that's why we count them as a C terminal end or the C terminal site. Now come here the beta sheet, we was talking about the beta sheet. So if both of the chain having the C terminal on the same side or the N terminal on the same side, we call them as a parallel. And if it is happened opposite, like N terminal and the C terminal are on the same side, that is the anti-parallel. Now these two are the basic. Other than there is a beta turn is the helix, beta turn also there, four amino acids are there. So like this they used to make the C terminal. So this primary structure, secondary structure, majority of the secondary structure is the alpha helix and the beta sheet. This alpha helix and beta sheet come together to make a tertiary structure, right? Like for example globulin proteins. So in the globulin proteins, 90% cases there is the alpha helix, and two alpha helix attached with this the beta turn. Now it is the tertiary structure, kind of a model swapping. So some like 5 alpha helix, 2 beta acetate, they make a type of a proteins. And another one, for example, if we take the protein channels that is present in the cell membrane, right? So in that cases there is also alpha helix and beta acid is also present. Okay, so beta acid also make a channel, the channel in the sense the gateway or the route through which any kind of a ionic charge or the ionic particle can come inside the cell. Okay, come here. So first one, primary structure, secondary structure, tertiary structure, and the quaternary. Quaternary structure mean the sense if the tertiary structure, three tertiary structure came together to make a quaternary structure. Very good example is the hemoglobins. There is a alpha to beta 2. That means I'm not here. It is the helix, not a helix. Okay, there is a alpha chain. So alpha subunit, not a chain, alpha subunit. Okay, two alpha subunit, two beta subunit. So make together they make a tetramer. Okay, so it is a, if there is a both of the tertiary structure are the same, then we call them as a homodimer. If it is a different, then we call them as a heterodimer. Like for example, if we consider the alpha and the beta subunit, so these two alpha and beta subunit of the hemoglobin, they are the heterodimer, and the alpha alpha subunit, there is the homodimer. So this was the primary structure, secondary structure, and the tertiary structure, quaternary structure. Primary structure nothing but arrangement of amino acid. Secondary structure, it is the alpha helix beta acid. Tertiary structure, this all the secondary structure come together to make a three-dimensional structure. We call them as a tertiary structure, and this tertiary structure come together as a subunit to make a quaternary structure. So this was the structure of a protein, very basic and very easy.